Hello. Am I in the picture somewhere? Can I wave to you or? Yep. Hello, everybody. Great to see you. Um, I will try to start as quickly as possible now. Everything should be ready. I hope you see there's a meeting invitation, something. Do you have that on your screen, all of you? Yeah? Okay. So I think I will just begin. We don't have very much time, so I'll try to go fairly quickly through everything. So, oops. Yeah. So this is somebody else is coming in. Goodberg, you see that? You need to admit somebody there.
Yeah, I will be monitoring this.
Okay. It's right above my slide here. Anyway, the title of my talk is From T-Patterns and T-Strings to Genes and Curricula and Human T-Societies. And recently I definitely realized that these two T-societies can be called the most complex structures in the known universe. One might think this was a terrible overstatement or something like that, but I have tested in a few scientific contexts, and I think myself it's obvious, but we will see. Presentation overview. Origins, ethology, and the search for human uniqueness, 1960 to 1970s. T-patterns, hidden temporal patterns in human, animal, and neuronal interactions. T-strings, spatial T-patterns on physical strings from DNA to texts. T-societies, two types of mass societies, interactive emergent societies and T-societies. I will, of course, explain that, define that, whatever we call that. And this thing about human uniqueness. There is an explosion in human social organization since about the 800s, and there is now a very striking, in my mind, a biomathematical self-similarity, which humanity has never really experienced until so recently. When I started looking for human uniqueness, I was actually expecting the opposite. I thought when I learned about social insects that it would turn out that we would be pretty much the same. We are so similar in many ways to primates and so on. And when we went to mass societies, which is totally unknown in other primates, when we got there, I thought we would maybe be pretty much like the social insects. And even if there is some truth in that, it's in one way extremely different. That was the biggest surprise of all for me. Okay, origins, ethology, and the search for what makes human societies unique. Oh, I don't see the top of my slide because of something here from, Okay, I think I got rid. So, searching for uniqueness of human mass societies, work inspired in the 1960s by the biology of behavior, ethology, of Desmond Morris and Nico Tinbergen. I read these in the middle of the 60s. Tinbergen shared the 1970 Nobel Prize in Physiology or Medicine with Konrad Lorenz and Carl von Frisch. Key questions: what suddenly made advanced modern human societies completely superior in countless ways, so different from anything else in nature? So you see what are some basic writings, which shows also that I've been working on this for over half a century. See, it's communication and social organization of social insects and primates, humans included. It was a BA thesis, a very highly motivated 80-page thesis. I think it was really like the program for my life. And then I had this medal thesis at the University of Copenhagen, 79. And then finally I gave a talk, my first talk outside the university in 1982. I called it temporal configuration analysis, detection of inequal dynamic structure through artificial categorization of a real-time behavioral stream. It was a workshop on artificial intelligence long before that was a big subject. And at that time, the computers were not ready at all, and the internet wasn't either. There was not this massive access to human writings, and the computers were about 60K, not 60 giga, but 60K. Okay. Yeah. Here is a concept that for some are not totally familiar, so I just refresh that a little bit. Self-similarity and the biology of behavior. Self-similarity, when a whole and at least one of its parts are similar. In 1973, when Tinberg, Lawrence, and von Frisch shared the Nobel Prize for ethology, there was much talk of similarities and analogies between species. Lawrence's Nobel lecture was entitled Analogy as a Source of Knowledge. But the smallest species studied were insects. None were parts of others. No species was a part of another. There was no talk yet of self-similarity. Studying social and biological patterning. This is for my work was first decades focused on hidden temporal social interaction patterns, first in humans, children, and other animals, then in brain neuronal networks, then analogous patterns on strings from molecules to texts, social organization emerging from individual interactions and T-pattern strings. Contributions since the 70s. It's my work there. Since the 70s. Research and development. T-pattern and T-system model creation. Detection algorithm development. Theme software from 3,000 lines on PTP 11 64K to over 300,000 lines today. Actually, it would have been more like 400,000, but I am reducing the size, making more compact, so it's probably more like 150,000 lines now, and much more easy and compact and concise, easy to maintain and develop. I've been working, and has been much more work than expected, much harder than expected, to create parallel processing T-pattern detection software. I have a 32-core CPU desktop, and I also have on that one a GPU, which you probably know, but we can, if you want, we can discuss that at the end. These two are now working since a week in such a way that they give exactly the same results, even if they are really very, very different. Then I've been doing with the collaborations with many people. Some of you are there. T-pattern detection in behavior and interactions in humans, behavior and interactions in animals, neural networks in brains, DNA and protein structure, spatial T-strings. Key publications, some of you are very familiar with and may have participated in. We have for a long time now, there's a formal inter-university research network since 95, really, 1995, of 38 universities, some say 40 universities, formed around T-pattern analysis. Then we have these books. The first one, 2005, The Hidden Structure of Interaction from Neurons to Culture Patterns. That's with Anoli et al. Then in 2009, I participated in this book about the biology of religious behavior, which is a strong interest of mine. And then in 2016, it's a Springer book discovering hidden patterns in behavior and interactions. There were decades of collaborations with the University of Chicago, University of Paris, University of Barcelona, University of Cambridge, UK, and the Museum of Natural History in Paris, where I worked for 4 years in anthropology, subdirector. See also comprehensive review by Casaruba et al., 2015, Journal of Neuroscience Methods. T patterns, hidden temporal patterns in behavior and interactions. This is the view when I was beginning to analyze children's interactions. Oops. I was very much inspired by the work of Hubert Montagnier in France, studying interactions both in social insects and in children. But it was clear that something more was needed, and the computers were getting online, and there seemed to be a possibility there. I tried first to work with the IBM Center, the statisticians there. I was lucky one of my countrymen was heading that, and but it came clear pretty early that the ordinary multivariate statistical methods, they didn't cut it. They were not made for that. They were not made for data as you have in front of you now. Point series, lots of point series, where each, for example, here is the occurrence of some behavior, beginning or ending of some behavior by somebody. And there were many different types of behavior. In this case, 82 ethological event types. So it was just the next decision was, should I give up on this? Because there was no software that I could use, and I was not going to program myself very, very hard, and I didn't think I had the patience for it, and etc. But it turned out to be the only way possible. So we have this data, and how can we find sequences in that? And mostly at that time, people were looking for sequences of interactions or simply in individuals, and it was not going very well. They at the most found sequences of a few behaviors, and they were of questionable quality. So I ended up with this, what I call now this T-pattern model, and algorithms developed on these PTP-11 computers in Fortran 4. And to make this very short, when I finally applied that to exactly these data, everybody was surprised what was found. I'll just show you. This is what I call a pseudofractal self-similar object. The T-pattern is a series of behaviors separated by characteristic approximate intervals between them, distances between them. So each X is an event type or a T-pattern, can itself be a T-pattern. This is hierarchical, recursive. The general term means after xi occurs at time t, significantly more often than expected by chance, xi plus 1 occurs within a critical interval. This t plus d1, t plus d2. Key property. Q can be recursively split into a statistical binary fractal tree of critical intervals. So Q equals Q left and a Q right separated by a critical interval, d1, d2. Where this tree, its terminals are then the behaviors of the patterns. Detection works in the opposite direction from the occurrence series using pattern completeness competition, which was essential, and evolution algorithms, which is the same, really. Otherwise, everything explodes because there's far too much redundancy in detection. So this is what turned out. This was found in the points that I just showed you before, the data. This extremely irregular repeated pattern just turned up. Here on the left, can you see the pointer? I assume you do. On the left, you have the detection tree. The gradual patterns of patterns of patterns of patterns. That's the static tree. Here you have the points relative to each one of these elements, these event types. And you can see how this pattern is repeated almost identically 4 times until the children stood up and they were free to end whenever they wanted. Actually, the last occurrence of this pattern is rather like slowed down, as if they were getting bored. Up here you see this as a dynamic tree. They represent better the actual structure of the tree here. While this is static, it showed the hierarchical buildup. Okay. T patterns in the brain, neural network interactions and cell similarity. Sometime, I don't remember any longer, it's probably around 2000, got in contact with people in Cambridge who were studying interactions of neurons in rat brains, olfactory bulb, and they had fantastic data collected with chips in the brain. It was very new, and they could register the average spike of up to hundreds of neurons, all in the vicinity of each other. But they had complete trouble analyzing these data. Brain is a self-similar structure, or as Olav Sporn says in this book, the brain structures here are hierarchical and self-similar. A nested hierarchy, smaller elements joined together to form larger elements, which in turn form even larger elements, and so forth. Many of the integrative aspects of brain function depend on this multi-scale structural arrangement. So you see this fractal structure, before we talk about the timing, is already there. So this is one of the many data sets we got from these chips, and they were always organized. The limit was 12 breathing cycles. The registration was in time units of, I think, 1/1,000,000th of a second, something like that, but that was not needed. So we reduced that resolution. But you can see on the top here are the beginning and endings of the breathing cycles. This is just one rat. And this is one of the simpler data sets, easier to present here. And as you can see, it has been published, and we also presented a number of times in neuroscience conferences. So now the big question was, are there T patterns in this? Are there patterns the same kind as we have between the children? This was anything but known. And they had been working with computer scientists who used multivariate statistical methods that in some ways resemble more artificial intelligence methods today. They are recognizing objects and comparing objects as a whole. But we tried, And after some failures, because we were not sure if we were finding fake patterns, or there was something wrong with the data or something wrong with the algorithms or what. But we found out in the end, everything was really okay, and the patterns detected, even if they were really abundant, they were not. They were simply that. They were as with the children. So T patterns in brain neuronal network interactions. You see, this is exactly the same type of diagram that you saw before, and it links every inhalation up here except one. The T-band did not capture that, but maybe that's just about setting the search parameters a little bit differently. But you can see on the left you have the different neurons. You can even see that theme detects bursts in some of the neurons. And you have the exhalations, inhalations, and this is all connected up for all the 10 of the inhalations. You can also see this is also published. This was a huge surprise. But that work hasn't continued as it should have because of totally irrelevant personal events, which if you are in research for some time, you discover is very common that some things that occur in the personal life of people has much more influence than anything else. I mean, grants not coming in can be quite serious, but if somebody suddenly decides to move to China because of a divorce, etc., etc., it can change a lot. So self-similarity T patterns between and within brains. Here we have the two brains, children interacting, two brains interacting, and they make up these patterns that we just saw. Then we go into brains, and it's not children, but rats, but much indicates that any brain, mammalian brain, we have a lot in common. Then you see neurons. So probably within the brains of these children, there are these neuronal patterns of the same kind. This is what we call self-similarity. T-strings, spatial T-patterns on physical strings from molecules to texts. T-strings. T-strings are material physical strings with T-patterns, especially the giant extra-individual purely informational T-strings of the purely informational molecules of DNA and of texts. Proteins as T-strings. 90%, it turns out, 90% of words in text in general, unknown patterns in proteins, that was a few years ago, are T-patterns. That is T-strings, call them T-strings because now they are on molecules, like a discrete scale, but the scale is like ticks on a molecule. So this is one of those that I just detected on proteins. Here you have different amino residues. Some of them occur in bursts, as you can see here, and get into some of them are really long bursts, like here, and you can see there are 3 patterns here. And now something very new in theme. Now it's possible, finally, what I've hoped for a very long time, is to see a color-coded presentation of these molecules. This is now a part of theme. Once you have detected a pattern like that, and we are talking about that pattern, one can have a color-coded image of it that you can rotate as you want and so on. And actually, it seems that these three patterns, they correspond to these three things here. And moreover, to my surprise, I asked for AI to tell me something about what this was. And that was really a surprise because I could expect anything, but I got a whole more than a 4-page, of descriptions and explanations of what this was, and it was very interesting. But sadly, I only have one head and 24 hours per day, so I haven't been able to look at this much more, which I really look forward to do. Tea societies, 2 types of mass societies and the uniqueness of humans. 2 types of mass societies, they're interactive emergent society, simply practically all animal societies, and human societies until very recently. There are just humans or ants, or there are chimpanzees, or there are rats, and they are so and so many together, and they interact, and then depending on the species, you will get a different type of social structure. But there is another type, which I call a tea society. It has the same thing here, but it has an external string. Huge string, giant extra individual purely informational T string, which decides the characteristics to a large extent of each individual. It's not just determined by its genes and interactions with others. There is this external string that comes in many sections and that decides to a large extent the characteristics of each of the individuals as they interact also. So it's found only, this type of society, T society, only found in cells in the DNA world and in literate humans who have also actually reached a high level of specializing education. And then we are just talking about some 200 years. Okay, wait a second. Did I look something else like that? Interactive medicine society. It really more or less repeats what I said. JBit. And here you can see forming citizens in a T society. In proteins, segments of DNA on top there, T strings themselves, or they're called genes, are processed in ribosomes to produce specialized proteins. In humans, below, segments of the textome, Curricula, are processed in schools to produce specialized human citizens, now increasingly including AI and robots. So it seems that this development that began about 5,000 years ago with the invention of writing finally also includes specialized education, which is far more recent, and you have this same pattern inside the cell and inside human mass societies. Totally impossible for other primates. The highest number of individuals in primate societies is about just over 1,000, and usually much more. Okay, then you can see also T society, including T societies, only found in humans. You can see here have the cell. It is a citizen in bodies. They're made up of cells, either humans or ants. They are interactive emergent societies. They don't have the T string. Here we have the ant is a citizen in a hive, and hives, social insects, don't have the JPIT, the external strings. But while humans just recently are now citizens in mega societies, They are tea societies, as we said, and they have the self-similarity with cells. It is the only animal species ever to have that. Only have we reached, it's only humans who recently have reached that stage in development. No other animal species, no other form of life on Earth has reached that. And what a coincidence that at that same time everything has exploded in human life, technology and knowledge, science, and global warming and collapse of the biodiversity and so on. Enormous changes. So just to repeat this a little bit, RNA, interactive emergence versus DNA, protein, T society. You can see it here, left and right. I'll go faster a bit now. RNA, animal and earlier human societies versus recent human societies. We have the interactive emergence societies on the left, and we have text-based human society based also on specialized education. Education until about 800 was not specialization, just general education, and only for the small elites. It was not general. Protein and human T societies, unique self-similarity. So we have, in both cases, we have this huge string on the left. We have the society as before, but now every individual is massively influenced by parts of this stringome. Animal maximum population size, I just looked that up. Primate maximum is about 1,000. Insect hive maximum size is about in a few millions. Protein and human T society population size. China and India is over 1 billion each. Tells us, and that is very, very new. Humans were only 1 billion in 1800 and far less before. And here we have within the cells where you have T societies. Before multicellular life, they were counted in billions. And after multicellular life, the maximum was maybe hundreds of billions. But only recently through these T societies, so absolutely new in human existence, with some 200 years, do we reach these population sizes that are characteristic for internal and cellular societies of proteins. And we have seen some changes. Around the year 800, the turning point in human history, mass education, mass specialization, and the population explosion. We went from 1 billion to 8 billion in 200 years. And before that, the revolution had been very, very slow. We will see. Human population size since the last ice age. You can see how this evolution is practically flat. It is around 800, where really it grows very, very fast. See it again on a smaller scale, more clearly. If we go back to where writing was invented, about minus 3,000 years, we see nothing really big changes until 1800. Apart from there, then there is the population explosion with all kinds of other things going with it. And we take the last one, if we say from the printing, if printing was so important, but you can see there was no big jump there. The jump begins really around 800 with specialized education. Most people before that were either they were a part of the ruling elite or they were farmers and they could learn from each other directly. Since 1800, mass specialization. Before 1800, literacy rates were below 10% across Europe and even lower elsewhere. Education was not designed for professional specialization. After 1800, the transformation of education changed its very purpose, and brought it for the first time to the masses. This was arguably more significant and lasting than the mechanization of manufacturing. This transition coincided with a turning point in human history, an explosion in global population size, and several other key parameters. And we can just for the moment also talk about global warming and collapse of biodiversity, not to mention the rise of science and technology. And now it seems that very soon the most intelligent beings on this earth will be machines. Modern human uniqueness. Human T-society self-similarity with protein societies has only been observed in modern humans for about 2 centuries, making its sudden appearance unique and a turning point in human and Earth's natural history. Why say Earth's natural history? Because we are changing Earth's natural history with the collapse of biodiversity, with global warming, and effects of global warming, and it's really these two are really consequences of human population explosions. All this humans earlier were just far too few and had far too little technology to be able to create such problems. At the present crossroads, humanity has assumed that life's evolution has been self-organizing from the beginning, except those who believe there was a God there, some very recently. And the recent discovery of T-society self-similarity across billions of years suggests that this process is still ongoing. AI and robots, new citizens in the T-society. A T-society is a society of multiple kinds of specialized individuals defined by a J-bit. Human T-societies now increasingly include AI and robots as specialized citizens. Their behavioral potentials determine the segments of the taxone, just as human specialists are formed through curricula. It dawned on me not so long ago this really clear analogy between genes and curricula. It's rather striking. AI's use of JPITs is growing exponentially, but written formulas and prescripts replaced thinking long before computers. People had the Pythagoras rule long before computers and had them and used it instead of trying to think the whole thing out. They had a formula ready. This is not a break from the T-society pattern, it is its continuation. Viruses collaborating and competing texts. Just as molecular viruses exploit cells, textual T-viruses exploit the similarity of patterns in texts and brains. Fake superstitions or holy texts can be understood as T-viruses, JPITs that co-opt their society's mechanism for forming and directing citizens. Mathematical regularities in human social behavior, including religious behavior, have appeared and are already stored in JPITs. The T-society framework provides a new lens for understanding how such texts propagate and persist. And so this is the conclusion. The brain sometimes is considered the most complex object in the known universe, and I've heard that, I don't know how many times, hundreds of times. You would easily find that among people with medical education and so on. And I even recently saw a citation from one of the most famous physicists in the world who lives in New York, and he claims this, that the brain is the most complex object in the known universe. They sometimes say that culture for humans is like water for fish. They don't see it. They're just totally a part of it. And living in New York and working in New York and being totally a part of that system, he doesn't see that he is a part of a T-society that connects millions and tens of millions, and actually much more, of highly specialized brains which each one is the most complex in the world, according to him. So I think it's really reasonable. I thought so for a long time. That's realizing that societies are the most complex, human societies are most complex in the universe. And this is so massively underlined with these, when we understand that we are the first T societies in the universe, known universe, and not to mention in the evolution of life and Earth. Okay. These are key references if somebody is interested sometime. This is just at the end. It's too long to go through that here. We could quickly see maybe what's it, but it's all about what I'm talking about. Thank you very much. The human exception happened in a biological eye blink, from tiny communities to megacities, from stone axes to computers and spaceships. What happened? Unique biomathematical and social self-similarity. Thank you very much. I think we had agreed that it would be, I would tell something about news about theme. New version I'm still working on, and maybe questions and answers if somebody is interested. So, new features in theme 7, AI-assisted development. I actually think I might have given up on this if it wasn't that I have now learned to use AI much more and more effectively to help me with the work. Something that I did quite some time ago is fast data import. It was a problem in theme that importing data was only made for relatively small data. Now that has changed. Data import is extremely fast. Then it was another problem was that the analyzing and finding T patterns in very big data is very demanding in terms of calculation, in terms of computer power. So I started creating parallel processing where you have, for example, have a computer with 32 different cores, and I develop search engine that used that. But then came the GPU, what is now the main tool for artificial intelligence, and it seems that it's extremely well. Really, T-pattern detection is really naturally fit for parallel processing, and these GPUs have thousands of cores, not 32. Thousands of them. It's many ways quite different, but I've always worked in Delphi Pascal since my early Fortran days, but now I had to learn CUDA, which is the language for these GPUs, NVIDIA, and also C++, enough that they are very closely related. And only a week ago, these two engines, detection engines, very different in many ways, different languages. They gave exactly the same results. So that's comforting. I mean, exactly. And that is not something that I didn't know that until relatively recently, that in artificial intelligence is not only that they cannot always be sure, you can not always count on it's true what they are saying, but for calculations in science, you have to expect that there may be a variation, 1-2% imprecision. And nobody had told me, I'd never seen that anywhere, but recently I learned about that. And that's because I'm working on these GPUs. Okay, so there is the molecular protein analysis. Now I showed you before, we can have a protein and we could see the structure in terms of T patterns, and then we can see where in that molecule, that particular molecule in color-coded presentation, where these patterns are occurring. And you can also have simply sending that information to AI, and it will give you a report on what does that tell us? What are these patterns? Where are they? What do they occur anywhere? Do they occur across everything? Are they particular regions of interest? And it seems, I've just begun looking at it, seems that it very much are very clearly distinct areas, and they have something in common. So there is something that surprised me very much with these AI tools. I decided to give to Claude the paper about ThemeWatch, what it was about, and asked for help to develop it further. And it understood absolutely what this was about, found it very interesting, and asked if it could help me write a new version with a few other features that we had dreamt of adding, but it was too difficult. It took only 2 days to have most of the software to do this, have it written, both for iPhone and for Android phones, with lots of features that were impossible when I was doing this so and so in 2017 or something. And then I asked if it would, well, I think it suggested to make a user manual. Yes, it did that. Fine user manual. And also a technical manual. All this was with my left hand over 2 or 3 days. And now it's just that I am now I should finish a paper for a mathematics journal for the end of this month. And I don't have any time to finish this. It's a shame not being able to have collaborators that might be interesting. I think it would be, people could start a whole company around just that. There's so many possibilities in medical importance or clinical psychology and so on, and we could talk a lot about that much better another day if you want. But this is what's really coming up in theme 7. I don't think this molecular thing that's already integrated, the parallel detection, as I told you, the two detection engines are giving the same result a week ago. And so maybe the last thing I've been doing here is there have been a company to create SciPods and SciTubes, and also open access government to ask me for papers, and still does. And I am writing some of this, but if you like, you could find this SciPod, which is it's auditive, and SciTube, which is a video based on my work. I think they did that reasonably. I'm just an avatar there. It's not you don't see me, you see they have an avatar. And I think that's it. I think this can conclude our session. And if you have any questions, of course, I would be happy to answer questions or comments. That's fine.
Thank you very much, Professor Magnusson. We have already one here in the chat box from Canada. Thank you for a wonderful presentation. When can we expect theme 7 to be available?
I don't like making promises. It depends on various things. It depends on how with my collaborators we could do this. There is something called beta versions, and we might make, or I might make a beta version that would be fine for somebody, even if some other things in the beta version were not ready. So it could be a partial beta version, but software is updated very differently now than it was before. You would have a new version with everything complete in a year or 2 or 3, and then the next update would be in another 2 or 3 years. This has been the model. But now I'm working with one tool from Microsoft called Visual Studio, and there is Visual Studio Code, and there's Visual Studio Code Insider. And that's what I'm using, and they update practically every day. It's a development tool. So this thing about everything coming just one version per year or per 2 years or 5 years, that's finished. Everything goes so much faster today. Artificial intelligence just 2 or 3 years ago is totally unrecognizable. Okay, so there might be beta versions very soon, but maybe the complete version, if there is something like that, would be later. But maybe there will never be anything but these gradual additions and updates and so on. And with help of, I would find it extremely difficult after having worked on this for about 60 years now, if I didn't have this artificial intelligence assistance. But it's also a little bit of extra work because you have to know, you have to learn how to use it and how to escape the errors it makes for you. And that can sometimes be costly. But maintaining, for example, the software is absolutely invaluable to have AI tools to help you with that. There is a bug, it can find it, and it often can just solve it without you coding anything. And since about a year, I have hardly done any programming, always through AI. But you have to, as if I suddenly had a programming team. But you see, just following up on what AI just did for me with the theme watch, you ought even to just follow up on that and exploit that and get it out. I don't have the time now to do that at all. I must give priority to this mathematics paper. I just gave a talk a little over a month ago at the University of Applied Sciences in Berlin, that you have to prepare that. That actually went surprisingly well. That was a conference in material sciences, and I was just proposed that I would be a guest editor in biomedicine journal, etc. But this is just, time is so limited, and you have to choose what to do. That's why I cannot make very many promises, because everything is moving in a way.
Yeah. One more from the chat box from India.
Yep.
Our society adds more AI, like companion animals or digital assistants. My question is whether humans can also form T patterns with non-biological entities in the future.
Well, that's what we have already seen, and I think you are involved in that, Gudberg, with the.
I was, yeah.
Yeah, and that's with the people who work in robotics and robotic dogs and so on, and it actually, I wrote that somewhere more than 30 years ago, maybe more than 40 years ago, that if robots are going to be successful in human societies, they will have to make the same kinds of patterns and also to understand these patterns. And timing, I think we're just finishing a paper with Maurizio Casarrubia. Timing is in human interaction is really everything. If you say hello to somebody and that person either says hello much too fast, oh, you wonder, is something wrong with that person? Or it says hello, but after a second or two, which is far too much for normal people. So you would start judging people on such a basis, a tiny fraction, or even a fraction of a second might change your mind about that person. That's quite crazy. But probably if there is such a hesitation, then maybe that person is crazy. So what a problem. So I think maybe I answered the question of that from India.
Yeah, we have a raised hand from Stonebrook University.
Yeah.
Go ahead. You're on mute, so unmute yourself, please. Okay.
Hello, and thank you very much for organizing this presentation. I'm working in the field of neuroscience, broadly understood. To be more specific, I work on studying animals' behavior and neuronal circuits. Maybe you remember at some point I graduated from Dr. Konstantin Anokhin lab in Moscow, but it was many years ago now. So actually, from Irina Zarayskaya, I know about your work, and I'm curious. We are currently accumulating rather big data sets, both on animals' behavior, but not in real time. But we have snapshots. We know how they interact socially. We study intermale aggression. We also collect data, again, in the form of snapshots of their activity, of their neuronal activity. Specifically, we are looking at expression of a particular molecular marker in their brain. So I'm wondering how theme and in general your approach may help us to what predictions it can make if we find some patterns, let's say, in these dyadic animals interactions, or in how neuronal circuits are organized.
Oh, that's a huge lifelong project you have there. Judging by my experience. But these are probably all very interesting questions, and maybe I have something to help with. I can help in some way, but it will be too long for a discussion in this meeting. And what I suggest maybe is something that I will start doing. Things are changing so fast with this AI and so on. I'm telling you about it. For any of you, if you have such questions and you would like to discuss them, just like this, you just send me an email and we can agree on a time when we could simply discuss like we do here. Just with Teams or Zoom or anything else you like. Team seems to work very well. And so just send me an email then, and we agree upon on some time we can really talk about it. This is too complicated to discuss
I
in front of
understand.
everybody.
Yeah.
Sounds
good.
Great.
Yes.
It's great to hear that you were in Moscow.
I was in Moscow. Yeah, I graduated from there and I moved to the United States in 2016 now. But I'm from Dr. Anokhin's lab.
Oh, no, yeah, exactly. It's great to hear from you. I haven't heard from him directly or indirectly for a very, very long time.
Right.
Now, we got together very easily because we both come from chess-playing nations, and when we sat down to talk, it was as if we were sitting. We're going to play chess. It was very easy.
I understand.
Yeah.
Right. Thank you very much.
Give my best to him if you have any contact.
I will.
Yeah.
Thank you. Thank you.
Yes. Yeah. Bye. Yes.
All right. One more from the chat box. Daniel La Presa from Spain. He was having difficulties with his sound, so he didn't hear clearly what you said about when Theme 7 will be available. And he's also asking, will there also be an edu version?
Oh, yeah. I definitely think there would be an edu version. But now we can also think about having a version which does not have parallel processing. We could have another one that has one parallel processing, and another one that has a more powerful parallel processing. But I haven't had any time to think about these things. We have to think about it, but I would always want to have versions that are enough for students. But if people who are doing some really serious research and need some serious processing power and so on, it would be very nice if I get some economical help out of that, because I work with no resources except myself.
All right. Any more, maybe question? One more question, if anyone, or comments before we close this meeting? No one has raised a hand. Magnus, anything more from you?
I think I've already said quite a bit, so I think I should stop now.
All right.
And I was very happy to see you, and very thankful that you would come by and listen, even participate. Thank you very much indeed.
Thank you very much, everyone, for joining us today. I will be sending you a copy of the presentation, and if the recording is of good quality, we'll make that available to you within the next few days. Thank you very much. Have a good day, evening, night, wherever you are located on the globe.
Thank you.
Thank you.
Thank
Bye-bye.
you.
Thank you.
Bye.
Bye-bye.
Bye-bye.